Hi everyone, welcome back to Biology 242. This is the second part of the blood lecture. So where we left off, we were talking about the disposal of red blood cells, specifically as it relates to how hemoglobin is safely processed and excreted. So remember, although hemoglobin is great when it's inside of our red blood cells, it does very, very bad damage if it is allowed to bop around in the plasma outside of red blood cells. So those consequences include things like blocking kidney tubules and causing kidney failure, or causing other ischemic events in other parts of the body as it blocks small blood vessels. So hemoglobin, great inside, not great outside. Alrighty, so now we're going to get into erythropoiesis and revisit some of that stuff. So your education about erythropoiesis probably started when we talked about the the skeletal system. So it was addressed briefly in the context of talking about bone marrow, but then we kind of left it for later. So now we'll talk about it some more. So erythropoiesis is red blood cell formation. It seems appropriate to get a reddish pen out, so let's do that. And this is specifically going to happen in the red bone marrow of adults. And assuming all of the levels of erythropoietin are normal, you should be making about 3 million red blood cells per second. So what this ends up meaning is you're replacing them at about the rate they die. So a replacement should approximately equal destruction. So in adults, like I said, red bone marrow is where this happens, and the red bone marrow that you have left over by the time you are fully grown up is limited to a couple specific spots in the body, so not everywhere. If you are a fetus or an embryo, I assume nobody watching this video is a fetus or an embryo, then you don't have fully formed bones yet, so the yolk sac, liver, spleen, and whatever bone marrow exists as you develop will perform that task. So the stimulus to increase erythropoietin secretion, which is, by the way, kidney hormone, and therefore increase the production of red blood cells is hypoxia. So any transient drop in tissue oxygen is going to stimulate erythropoietin secretion. Okay, so we did address this a little bit. I'm just coming back to it because it's been a while. So hemocytoblasts are cells that can become a blood cell, and it even says that in the name. So here we have hemo, cyto, and then blast. From there, we've got two different possibilities. One is these cells can differentiate into lymphoid stem cells, which would become lymphocytes. The other is differentiation into myeloid stem cells in response to a signaling molecule called multi-colony stimulating factor. And these guys have a lot more variety as far as what they can differentiate into. So they can differentiate into the red blood cell tract, which is here, or into megakaryocytes that become platelets, or the set of cells that become either granulocytes or agranulocyte monocytes. So it's weird that the agranulocytes have two different lineages, whereas all of the granulocytes come from the same place. So just to go over sort of the specifics of erythropoiesis, myeloid stem cells, in response to erythropoietin, become pro-erythroblasts, and those are continued to be stimulated by erythropoietin. So what you're seeing here is this increase in reddish-orange color. So basically, while this cell still has a nucleus, its job is to transcribe and translate the genes that code for hemoglobin as much as is possible. So basically, the cell's job is to just fill up with hemoglobin as much as it can before it spits out its nucleus and basically eats up all of its organelles. So what you get at the end is this little sort of discoid bag of hemoglobin, and that's what your red blood cells are. So here is another summary of the same thing. So we've got pro-erythroblasts, and those are basically precursor cells. And then we have erythroblasts, which again are going to produce hemoglobin. So like I said in the last slide, the cell's whole job, while it still has a nucleus, is to produce hemoglobin before the nucleus goes away. So reticulocytes are called that because they have this reticular network of degrading organelles. And these enter circulation. So if you look at a human blood smear, a very small percentage of the red blood cells that you see in that smear are actually going to be circulating reticulocytes, and they mature while they are circulating. So it takes about 1 or 2 days for them to fully come into erythrocyte form. And again, as I said in a previous slide, assuming everything is normal, erythropoiesis and the destruction of red blood cells should occur at the same rate. If destruction outpaces erythropoiesis, you get anemia, and if erythropoiesis outpaces destruction, you get polycythemia. Neither of those things are good. You want a happy medium. Okay, so requirements of erythropoiesis, meaning what do you need to build good blood? Well, first of all, if the whole point of having red blood cells is hemoglobin, you need to be able to build hemoglobin. So that's intrinsic in building red blood cells. So this is going to require iron. And primarily this iron comes from diet or recycling. So as we discussed previously, during the destruction of hemoglobin, the iron is transferred back to the liver and bone marrow by transferrin and other iron transport proteins. So some amount of iron is recycled, but you still need to eat plenty of iron. So dietary iron is still important. And of course, you also need amino acids to make the globin portion because it's just a protein. It's a tetramer, which means that you have to take 4 chains of amino acids, fold them up, and stick them together. You also need vitamins, so B12 is one, and this is important for protein synthesis and a little bit for iron homeostasis. B6 and folic acid, so all of these are going to facilitate and support this. Interestingly, you cannot absorb vitamin B12 without intrinsic factor. An intrinsic factor comes from enteroendocrine cells in the gastric mucosa. So the only part of your stomach that has an indispensable or irreplaceable function is actually the enteroendocrine cells that produce intrinsic factor. So you can have a total gastrectomy, and as long as you have supplementation with intrinsic factor, you should be fine. It's just that vitamin B12 can't get into your bloodstream and be absorbed without intrinsic factor. Erythropoietin, which is abbreviated EPO, and other hormones stimulate erythropoiesis. So T3 by modifying metabolism, so increasing ATP production, and then androgens and growth hormone also spur on erythropoiesis. So androgens are why there's a little bit of sexual dimorphism in hematocrit between genetic males and genetic females, and then growth hormone because obviously if you're growing, you need to keep growing blood to keep up with the increasing size of your body. Oopsie. There we go. Okay. Blood typing. Blood typing gets really tricky. And the reason for that is that you have to keep 2 things in mind at the same time if you're going to show mastery of blood typing. So one of the things that you're going to have to keep track of is the surface antigen on the surface of the red blood cell. So at the end of the day, red blood cells are still cells, even though they lack all the component parts that we think of as being cell-ish, like nuclei. Excuse me, I was suppressing a sneeze there. So they have a phospholipid bilayer for a membrane just like any other cell, and in that phospholipid bilayer they have cell surface marker proteins, so proteins that stick through the membrane that have little branchy bits hanging off of them, and those are generally for basically identification. So you can think of it as being sort of like the cell's name tag. So all of your cells have surface markers. So, for example, if I took one of my skin cells, I could examine its surface and I would find that it has surface markers that correspond to my specific immunochemistry. But blood cells have some special additional ones. So, we call these antigens. And antigen is defined as just being any substance that is capable of eliciting some kind of an immune response. So, our immune systems are not supposed to respond to our own antigens. We call those self-antigens. Our immune system is supposed to respond to antigens that are quote-unquote not self. So that's the difference there. And these antigens are genetically determined. So you can actually use Punnett squares to figure out the likelihood of having a baby of a particular blood type. So for example, can do / predict Punnett squares. Remember those from Biology 160? Yeah, they're back. So the other thing that's tricky about blood typing is the antibodies in the plasma. So antigens are stuck onto surface of cells. That's what this picture is showing. Antibodies are non-living. These are plasma proteins. We also call them immunoglobulins. I almost said immunoglobulins, which is really funny. Not those. There are no goblins in your blood, at least as far as I'm aware. So immunoglobulins, these are Y-shaped proteins, and they have a specific part, usually on the end of the Y here, that has the appropriate amino acid sequence to stick to something. So specific to a particular antigen is the way I'll say that. All right, so there you go. So this antigen-antibody combo is what makes blood typing really tricky for students. And I hear this over and over again. Students will study this for a while and say, okay, I think I get it. And then go to the test and then come back to me after the test and say, wow, I thought I understood blood typing, but it's clear that I really don't. And the reason for that is it's easy to go, for example, Google blood typing charts and just try to memorize the chart, like who can transfuse whom or whatever. But that doesn't mean that you understand the underlying physiology of the reaction, and that's what I'm going to ask you about on tests. So if you have this impulse to go like on Study Blue and be like, let's memorize this entire blood typing chart. If you want to do that, that's totally fine. But be aware that that doesn't really engender critical thinking about blood typing and blood serology, so make sure that you're also practicing. So practice with your blood types, and I'll model that for you as well. Okay, so you have two major antigen groups, and these are ABO. So you have A, you have B, you have both, or you have neither. And RH, which is also sometimes called D. So I want you to know these are equivalent. This is another thing that students sometimes miss. Like if I put anti-RH in a question and they're like, well, I thought it was D, or I didn't realize this was D. So make sure that you know these are equivalent terms, and I can use either. So you need to be prepared to interpret them. So we're going to talk about ABO first. And also, there are other minor antigen groups, about 20 or so that are known. We're not going to talk about any of those. That's for more complex serology that we just don't have time to get into in this class. So, oops, excuse me, I didn't mean to tap the mic, I apologize. I was trying to move it, so I am sorry if that was loud. So what antigens do we have? Well, A, B, both or neither, as I said. And this chart really nicely summarizes. So this row is what's on red blood cells. This row is what's in your plasma. So someone with type A surface antigens is going to have antibodies that are supposed to attack type B surface antigens. Somebody with type B surface antigens has antibodies that are going to attack type A antigens. Blood type AB has both, and blood type O has neither. So when I say both and neither, what I mean, AB has both A and B surface antigens. Type O has neither surface antigen, but we have this weird cross sort of or reciprocal situation where no antibodies, at least not against blood. Of course, you have to have other kinds. And in type O, you don't have any surface antigens, so you have all antibodies possible. Okay, so let's move on from that. So here's where it starts to get tricky. If you are a clinician, one thing that you may end up doing is performing or assisting with transfusions. This is why blood typing matters. So essentially, if you go, let's say you go to the Red Cross truck and you decide to donate blood, one thing that's going to be necessary is they're going to make sure that your blood type is known, and if you don't know it, they'll test you. They test it anyway. And that's so that they can avoid using the blood that you donate and transfusing it into the wrong person and causing that person's severe illness or death. So when foreign or non-self cells are introduced into the body, in this case we're talking about blood cells, antibodies are going to bind to the antigens on the non-self cells. So this results in something called agglutination. And it's also called a transfusion reaction or a cross reaction, just meaning that the wrong kind of blood was inserted into a patient and the patient's own plasma antibodies attacked the donor blood. So let's write that down, because that's also something that's not often clear to students. So patient/recipient, their antibodies attack donor blood cells. And I want to stress this is not the other way around. So one thing that students sort of get hung up on, and I understand why, because it's confusing. I've had students after tests say, oh, I thought that it was the donor's antibodies attacking the recipient's blood cells, and no, that's not the case. So one small detail to remember about this transfusion stuff is that when transfusions are given, the immunoglobulins of donor blood do not factor in to the transfusion reaction. So that's an important thing to keep in mind as well. The reason that, we might consider this is because, if you consider things like universal recipient or universal donor, the universal donor, for example, so let's say you're type B and you need blood and there's type AB or type O available. Well, type AB is half of the requirement, so you have B, right? But you also have these pesky A antigens, and those are going to screw things up. So your only real option is O. So if I put O red blood cells into B blood, the anti-A antibodies won't stick to anything on the surface of those cells, so we're fine. But if I gave whole O blood, then these anti-B antibodies would stick to this person's surface antigens. The reason that's not the case is because donor blood undergoes some processes, including plasmapheresis, that get those immunoglobulins out so that they don't affect the patient's or recipient's blood. Okay, so why is this bad at all? Well, similar to how it's bad when hemoglobin is out of red blood cells, this clumping that you see due to agglutination, so that's pictured in this image right here, this clumping creates little rafts of erythrocytes. And if you think about what we discussed with red blood cells and how they form those little rouleau stacks and they can flow through capillaries one single file at a time, these big old clumps cannot do that. And so they're going to get to the entrance to a capillary bed and basically block it. So blocking small vessels is really bad for organs and tissues because it causes ischemia. And ischemia is the medical term for a lack of blood supply to a particular area. So localized ischemias cause localized tissue death. So these are specifically risky if they happen at the brain, heart, or the kidney. So again, renal failure, stroke, or myocardial infarction are the major risks here. Okay, so plus or minus. This is referring to the either presence or absence of the Rh antigen. Remember, Rh is sometimes called D, so these are not different things, these are the same thing. So, another thing that is difficult to get your head around if you're just learning this stuff is that the Rh antigen is independent of the A, B, or O. So, for example, what I mean when I say that is if you have blood type B, that doesn't predict anything about what your Rh type will be. So you could be B positive or B negative, and that's genetically determined as well. But those two genes do not display any linkage traits, meaning that the presence of one does not affect the presence of another. So this is where we get the blood types A positive, A negative, B positive, B negative, etc. So with the Rh types, there is a specific problem, and that is the following. So I'm going to actually do some board work for this. Let's use my blackboard instead. There we go. So this is a crash course on ABO versus Rh. So over here I'm going to write ABO. So antigens are inherited, meaning that they are genetically determined, and antibodies against non-self blood are present at birth. Now, interestingly, why did I make that Y look so weird? There we go. Let's try that again. So I was asked this by a student a couple years ago. So how are the antibodies present at birth if you needed an exposure event to make antibodies against something? And that's a great question. The answer was, at that time, gosh, I don't know. I just took that fact at face value. Since I've done some research and found out that actually nobody knows. So this is an area of science that is still a black box. There's still room for discovery there. There are competing theories. Some people think that environmental antigens resemble blood cell antigens enough that you get antibodies that work for both. There's other theorized mechanisms, but nobody has really nailed that down. So it's still an unknown as far as science is concerned. Interesting, eh? Okay, so let's switch colors here. So for the Rh group, wow, you cannot tell I switch colors like at all. Let's try orange. How's that? So Rh factor, in this case, antigens are also inherited. But the antibodies are not inborn. So what this means is you need to be exposed. If you are Rh negative individuals would need to be exposed to Rh positive blood at least once to gain anti-Rh antibodies. So, in summary, no exposure event required. versus initial exposure required. So that's a difference, and that difference has some consequences medically. So let's go examine those. Oh my gosh, a dog just barked outside, but it sounded like a human scream. It scared the living daylights out of me. My goodness. Okay. Okay, so that brings us to hemolytic disease of the newborn. So what I'm not going to do is read you these giant blocks of text because that's stupid. You can read those for yourself. I'm just going to kind of tell you the story of how this works while you look at the slide, because I think that it's easier to understand things if they're in a little bit more of a narrative format for a lot of people. So here's the deal with this. During pregnancy, and unfortunately we don't really get to talk much about the biology of pregnancy in this class. I wish we had more time for it because it's fascinating. We just don't. So you're going to get a little bit of a crash course on parts of it here. So during pregnancy, the fetus and the mom team up to make this incredible new organ called the placenta. So the placenta does a lot of different things. It's sort of generalized as being like the fetal nourishment doohickey, but it does a lot more stuff than that. So for example, it has tremendous endocrine function. It secretes progesterone to help maintain the pregnancy and estrogens and other hormones, as well as communicates between the fetus and the mother, and also provides an interface across which nutrients and oxygen can be exchanged between fetal and maternal blood. So, placenta, highly underrated organ, very, very cool. So people who don't have a uterus don't get this weird chance to make an organ from scratch while making a person from scratch, potentially more than one time per lifetime. There's other cool things about people without uteruses, but this is a specifically uterine superpower. So in order for the maternal and fetal blood supply to successfully exchange things but not actually touch, because we don't want separate blood supplies to touch, that risks transfusion reaction, they're kept separately but in close proximity. However, and I know many of my students are well aware of this because a lot of my students are moms and have had babies. So delivery is not a tidy process. After expulsion of the fetus, which comes out kind of covered in goo and screeching and reddish and whatnot, newborns are not as cute in real life as they are in the movies the minute they come out. But after that, there's still the task of delivering the placenta, which basically rips off the wall of the uterus, and that also rips blood vessels. So that creates an opportunity for maternal and fetal blood supplies to have contact. Now, typically this isn't an emergency, so the hemorrhaging is typically managed, but there is a little bit of blood mixing. So here's the rub. If someone who is Rh negative carries a child who is Rh positive, totally possible genetically, it's possible that the Rh positive antigens on the fetal blood will make their way into the maternal blood supply, or at least mix with it during delivery. And about 20% of Rh negative mothers who carry Rh positive children, due to that blood mixing, will become sensitized to the Rh factor within 6 months. And here sensitized means they will develop antibodies against Rh positive blood. So that's that exposure event I was talking about earlier. However, that's not enough time to generate antibodies that will affect the fetus. So that first Rh positive fetus, no big deal. Second pregnancy, however, this is where things become an issue. So if the Rh mother carries a second fetus and the second fetus is Rh positive, then maternal antibodies against the Rh factor are small enough to cross the placental barrier and go into the fetal bloodstream, where they will begin to stick to and attack the fetal erythrocytes, and that's bad news. So that causes hemolysis of fetal blood, which is why it's called hemolytic disease of the newborn. So this used to account for a fair amount of infant mortality and even sometimes spontaneous abortion in the ye olden times before immunology was well established. However, now we have a antibody-based drug named Rogam. And what this specifically does is this is a drug that's administered to a mother who is Rh negative during pregnancy and also after delivery. And basically, the antibodies in Rogam destroy the fetal red blood cells if they're present in the mom before they can stimulate a maternal immune response. So basically, they get to the fetal red blood cells first so that sensitization cannot occur. So that's the general idea. Now, if you're looking for a pick-me-up, because I know that the news is pretty dark these days, there is a man who has blood that contains the specific antibodies that RhoGAM is made of. So yes, this is a drug that's derived from actual people products, not invented in a lab. And this man's donated blood has been used to make RhoGAM that has treated hundreds of thousands of babies and saved hundreds of thousands of babies in his lifetime. So if you go ahead and Google like male donor RhoGAM or something like that, the article pops right up, and it's this charming old man seated in a chair surrounded by a bunch of really happy moms and happy babies that he helped save. So it's a feel-good story that's related to what you're learning. And I support anything that you can do that helps keep your morale high during these weird times we're living in. So this is just the summary of everything I just said. So again, not going to read it to you. It's just a recap of the story I just told you. So I do expect you to understand this for the exam, but you're going to want to practice with this idea, and I will give you opportunities to do that, as well as helping model practicing it for you during our Zoom sessions. So don't worry if you don't get it right off the bat. That doesn't mean that there's anything wrong with you. It just means that this is a really hard topic and you're going to need to work with it a little bit before it sticks. Okay, so before I move on to white blood cells, I just want to give you a little bit of an idea of what I mean by practicing. So right now I'm going to turn on my black screen and give you a little bit of an idea of like what a practice problem would look like, so that you know what to expect as far as like practicing for blood supply, or blood typing rather. So let's start with a really simple one since we're just learning this stuff. So let me think. So, pretty simple and straightforward. What blood types can an A-positive person safely receive? So in these typing questions, one of your keywords that you're going to need to pay attention to is whether I say receive or donate to. The reason for that is because in one case, I'm asking about the patient's plasma, and the other case, I'm asking about the donor's surface antigens, and you have to keep track of both of those. So receive versus donate to, those answers can actually be very different. And I'll show you that difference in a moment. So I like to do the problem this way. So I'm going to switch colors. So if this was my problem and I was faced with solving it, before I start thinking about donors, I'm just going to think about what this means. So what does this person's red blood cells look like? So I like drawing this. That's my personal preference. You don't have to. So I'm going to draw A antigens, and I've decided to make them blue because I decided to. And these are Ds. Now remember, the plus or minus of the Rh factor is it's there or it isn't. So there is no such thing as like D plus or D minus. Those aren't a thing. It's just either there's a D there or there's not. So that's what this patient's red blood cells look like. We also have to think about their plasma. So what's in their plasma? So in their plasma, there's not going to be anti-A or anti-Rh antibodies, because that would mean that their plasma would be attacking their own red blood cells. Instead, they're going to have immunoglobulins that are, I like to draw the antigen right on it, against B. So for this reason, we want to avoid giving them anything with the B surface antigen because that would cause a transfusion reaction. So what I like to do now, and this is just for practicing, I wouldn't expect you to do this on the exam, but it's good for modeling and for keeping track of all of the blood types, is I like to just write them out and then cross off the ones that are impossible to transfuse with this person. Okay, so my person is A positive, so obviously they can receive their own blood. Duh. Now, can they receive anything else here? Well, they could receive A negative. They can't receive any of the ABs or the Bs because that B is present. And they can receive O positive or O negative because, again, no B antibody is present. So if you're confused about A positive or A negative, let's look. So I'm going to draw myself an A negative blood cell next to this plasma antibody and have you look and see whether you think an immune reaction would occur. So we've still got those A antigens, we just don't have any Rh factor, which means still there's nothing for the plasma antibodies of the recipient to react to. So see how here I drew out all 8 blood types and then crossed them off and circled them? It's just a way to ensure that you're doing mental accounting and you don't miss any possible answers or give the wrong one. So yes, it's a little bit superfluous, it's kind of extra, but if you're just learning, it's a good way to keep mental track of what you're doing so you don't screw up. So that's the idea there. So let's change this now. And I'm actually going to use my eraser. Well, actually, let's do this instead. So now I'm changing the words. I'm changing the verbiage to donate to. So let's go erase some stuff and then we'll think about what that means. Okay. So again, let's write our blood types out over here. I almost wrote C because I was on autopilot, haha. Okay, so this is the plus row because I say so, and then this is the minus row, again, because I say so. So this is a little bit trickier, but we can get one out of the way right off the bat. So they can definitely donate to the same kind as them. Now, can they donate to somebody who's A negative? Well, here's the deal. We can't know for sure whether an A negative person's plasma includes anti-Rh antibodies or not. We don't know if they've had an exposure event. We have to assume that they have for safety. So that means that A negative is out. We also can't have them donate to anybody with B, right? Because B individuals are going to have anti-A antibodies whether or not the Rh factor is present. Now let's think about, can they donate to somebody with AB? So if you're AB, you don't have anti-A or anti-B antibodies because otherwise you'd attack your own blood. So an AB recipient can receive A positive blood because it's just one component of their cells. So let's draw an AB positive person's blood using the same, oopsie, there we go. So if you have AB positive blood, basically you have all the things on your red blood cells. Now remember, when we think about donations, that means that we are not factoring the donor's plasma into the mix. So we're only thinking about the recipient's plasma. So if an A positive person is going to donate to somebody, it's just that their A positive red blood cells are going to intermingle with the red blood cells of the recipient. And so that means that AB negative is possible. Or AB positive is possible. AB negative, however, no, because again, we don't know about the exposure status of this minus person. So we can't safely assume that they would be okay with receiving positive blood. They also can't donate to O positive because someone with O is going to be assumed to have anti-B and anti-A antibodies, and they can't donate to O negative because this person has all possible antibodies, we have to assume. So anti-A, anti-B, and anti-RH. So notice that if we change the verbiage to donate to, the list of blood types that are safe is going to change as well. So receiving and donating are not necessarily reciprocal, so you have to really keep track of plasma and of surface antigens to do this thing correctly. I hope that makes sense. If it doesn't make sense right off the bat, again, please don't panic. It doesn't mean that you're doing anything wrong. It's just that this stuff is counterintuitive, and you have to keep a lot of mental tabs on things at the same time, and it really just takes some practice. So as long as you're practicing with it instead of trying to memorize the blood typing cross-reaction chart, you should be okay for the exam. All right, so let's get rid of that. And this video is now about 40-something minutes long, which is where I'm going to cut it off. So the next video I'm going to make will be about white blood cells and clotting, and that will be up very, very soon, probably tomorrow, which is Friday. So I thank you for your attention and have an awesome evening.